Okay, thank you very much. So then now I'd like to go on to sort of setting the research priorities that we feel would get us to a solution as fast as possible, if there is one. And what I just want to do is I want to use the same setup that we used in the talks. So I'd start with the systematics session. We had some discussion in the beginning about, let's say, the species identity of the beetle that is attacking trees in Israel and here in California. And there was some suggestion that molecular diagnostics in these things may not work quite as well as maybe in some other species. I think that the results that the Israelis showed us from where the beetle from Sri Lanka couldn't survive on the fungus from Israel and vice versa, plus the big differences that were shown in the DNA sequences that were done by Paul. I think most of us, and correct me if I'm wrong, are now of the opinion that these things indeed are two different species, and that one of the research needs is that somebody will take a real thorough look at the morphology in order to figure out if there's any kind of morphological feature that can be used to distinguish these things, so that according to the taxonomic nomenclature, this thing can be properly described as a new species. Anybody comment on that? There in the back. Huh?
I said you kept looking at me, so I guess you would.
Well, I was hoping you would support my point of view.
No, I agree. I agree. I think the biological information we heard and the genetic information that you guys have indicates that there's something different here in California and Israel. And I think it's important to get to the bottom of that, to figure out if it's a new species, if it's an old species with already that has a name for it. But I think that's important, just to put a name on it, so we could then start, putting data to the species and how it behaves and everything that goes with it. So I couldn't agree with you more.
Okay, great. Mark?
I'm just wondering if we need to be more ambitious in our approach and maybe try and do more than just the one or two species description, maybe try and do a revision of some of these groups that they've poorly studied, and perhaps going even further to doing maybe proactive surveys in some of the areas of origin where these beetles seem to pop up and keep coming here. We always seem to get caught flat-footed, it seems to me. We don't know the species, we don't know the names, where they've come from, and maybe we need to be looking beyond our borders and being far more proactive in dealing with some of these obvious threats. Yeah, I know.
Well, but we are in the lucky position that we're having a lot of collaborators send us beetles, and what also came a little bit from the work Paul has been doing is that maybe, and there's no morphological evidence for this, but maybe these things are not coming from Asia like we think, but from Africa. And Diane has a whole bunch of specimens pickled somewhere from various places in Africa that maybe we could look into to see if there's something else there. But indeed, a revision of this group would be a good thing.
Well, I feel that we're a little hung up on the Sri Lanka because supposedly it's a T-shot whole borer. If anything, where Brown reported from Thailand a lot more hosts, including avocados, and I think Thailand could be a very good source of information because of the diversity of hosts that are actually fruit trees, not necessarily only ornamentals.
It's definitely a possibility, and we should look into it because it will be important to figure out where this thing came from in order to to go there and maybe see if there's happens to be some biological control out there.
Well, we could certainly pass a resolution at this meeting, since you mentioned Thailand, there is an active bark beetle taxonomist specialist, so we could make a resolution that Roger Beaver shall go and collect new samples. But facetiousness aside, that is one area where we have someone who could possibly provide material.
Yeah, I
So, I
think,
mean, there's a certain network that we may be able to tap, but I think right now Africa's a problem. We don't have nobody on the net.
yeah. Well, Bob already suggested that, that the taxonomist in Thailand should be involved, since he knows these things forward and backward, apparently. So I think we should do that. Yes?
Yeah, I had a different take on the systematics discussion. The take-home message for me from Yuri's talk was that it's a sort of a fragmented population of clones around the world, rather than one where there's gene flow between different populations. And to me, what would be more sensible would be to picking some kind of a barcode and determining the haplotypes of all the beetles around the world. And in that way, you'll be able to get some idea of the international movements of the different pests. And by having a precise identification of the haplotype, then you'll be able to more accurately relate some of the information that's coming, say, from Israel. For example, if the beetle here in California is almost identical to that in Israel, then you can probably make some good extrapolations as to what is happening in Israel could relate here, whereas in Australia we seem to have another 2 haplotypes of the beetle, and so our experiences in Australia may not relate very well to what you have here.
Yeah, I mean, I cannot agree more with you. And that's what Paul showed in his poster. And it's that the California and Israel populations are identical. And the ones from Australia, Papua New Guinea, Thailand, Sri Lanka, and Florida are the different from something more closely related to what we think is the T-short whole border.
So to me, morphology is not precise enough.
No, I agree. I mean, you're arguing with the wrong person here, but there is, if we want to have a name on the thing, which for a lot of issues, regulatory issues, for all sort of issues, it's an important thing. And so that's, I think, where we need to try to find any kind of morphological feature that may distinguish them, and then, everybody will be happy. You can still identify them molecularly and morphologically. So, but I agree with you. Just knowing, the DNA sequence tells you. Yes.
So at the moment, it's no longer Eulalia truncatus, it's Eulalia sp. We don't know what it is. I mean, that's something that's important to, if you could get that clear.
Well,
we're not calling it that
so
anymore?
what we can only say at this point, in morphology at this point determines the name of the thing, and the morphologically we cannot distinguish the thing from what is known as Eulalia fornicatus. So that, I think, would be the scientific name that we have to use, but we can, sort of make a sort of a slight detour and call this thing, instead of the T-short hole borer, the polyphagous short hole borer, to distinguish it from that other form. So,
Just one comment on the common names. We can't start throwing around common names in the entomological world unless they're ESA approved.
sure.
And that if somebody wants to put forward this name as polyphagous T-short hole borer, and that needs to go through some formal channels. And then there's going to be a lot of pushback because common names are different in different parts of the world, the same thing. People often don't like using them.
Can I just make a comment? I'm trying to take notes here. Now, if you see what I'm putting up here doesn't agree with the consensus, or you don't agree with what I'm putting down, make sure you point it out, okay? Because the concept here is we're going to have a record of what we decide collectively. We'll send it out to everybody. But if we're not getting this up here right, let me know.
Yes, Jocelyn.
At the to be a bit of a devil's advocate here, I think we're sort of getting slightly off track in terms
Yes.
of focusing on the immediate problem that we have here.
Yeah.
I agree with you very much that we need to have some name that we can call this thing, But in terms of figuring out, the worldwide patterns of species or clones or whatever, I don't really see that is going to affect the immediate problem here very much. There may be the possibility of biological control. I don't discount that, but based on previous experience with related and similar types of beetles where biological control has not been a very effective factor, I think that we need to just deal with the issue of giving it some sort of name we can work with and move on to things that are of probably a higher priority for dealing with the immediate problem.
Okay. I think we've laid out the way in which we can get the thing a name. Do you agree or not?
One more thought on that. I just wonder if, you have the insect name, of course, which is one thing, but then the scenario of the dying trees is perhaps a different thing. And when you're trying to communicate this to the public, it's really very important to choose a good name, to describe. And I think there's probably disagreement in this room about whether this would be considered a disease, a syndrome, a complex, that type of thing. Coming up with because you're going to need to communicate this to regulatory agencies and the public. And there's different implications if we're calling it the T-shot hole borer, or we're calling it some other thing, because the T-shot hole borer would suggest that it's simply an insect issue to the layperson. So just something to think about.
Okay. Okay, we got a question over there.
I was just going to support Jason very, very strongly on that point. And we had an analogous situation in Australia where we had eucalyptus rust introduced there, but the common name that was given to it was myrtle rust, and no one knew what a myrtle was. And therefore there was absolutely no public perception about the disease. But if it had been called eucalyptus rust, then people would have been jumping up and down, and you would have had all the environmental groups, but hardly anyone knows what a myrtle is.
Yeah. Mary Lou.
I'm going to jump on this same bandwagon and give the example that we have with citrus and citrus greening. When we did the initial messaging for our public outreach program, what was determined was in California, green means something really good and environmentally safe. So we were very careful in crafting our messaging. So that's why, even though it's hard to say Wanlongbing, we always refer to it as that. We refer to it as HLB because it's similar to HIV. You laugh, but this is the real truth. And as a plant pathologist, this was like a real epiphany for me. Yeah. So that would be one recommendation I would have too. The ambrosia beetle, that sounds so nice. That sounds like that really nice fruity salad. So I think you really do need to stick with something like a shot hole borer, because a shot rings out and a bore makes holes, and that creates a negative or a bad thing, especially in the mind of the public. Well, and I think you're going to need to really engage the public as we did with citrus.
Yeah, well, we discussed what to call this thing, and initially, so this thing was called the T-shot hole borer. Well, that doesn't really resonate here very well. We don't have much tea. So then we suggested to call it the avocado shot hole borer. Let's say the avocado industry was not really interested in that name. And so they felt, and we also thought, well, heck, this thing is attacking a whole bunch of different trees. Let's call it the polyphagous shot hole borer, one that attacks many different trees. And so that's, the reasoning behind our naming of the thing. So, yes?
The point was raised about ESA common names. As long as we put the application forward, don't get too far into the weeds about the doubts about the genetic strains and Californicatus. The fact that it is known in literature as the T-shot hole borer has absolutely no bearing on what we choose to propose to ESA.
Okay.
So as long as we keep the application clean, stay away from the genetics for now, call it fornicative, then we have a clear path to, and because I think what ESA looks at particularly, they don't like to see trivially, just because you have to write a paper that you need a common name.
Yeah.
So as long as you show some economic justification, I'm not going to really give you a lot of trouble, because they're going to send the thing to Bob or Bobby or to me or someone else, and people we know. So I think we made, just as a strategy.
Okay, sounds like a plan. Mark.
I've just been devil advocate, but like Jocelyn is, if we're dealing with the public, do we really want to name it polyphagus?
What do you want to call it, Mark? If you have a better suggestion, eats many things.
How about the Greek shot hole borer? Then we need a smaller name in front of shot hole borer.
Omnivorous.
Yeah, omnivorous was what was suggested initially, but we had a linguist in our committee that decided this, who pointed out that omnivorous means something else than prolificus, and prolificus was the more proper version of all of this. Anyway, I think we have an issue with this name. Yes, Akif.
I comment on the disease name part of it. So when we first found this one, because of the symptom that we have seen on the avocado, we start calling this one Fusarium dieback and shot hole borer. So actually we published a disease note about that one too. So that's something that we need to talk about, like, are we gonna call the complex as Jason's recommend, or just give the one disease, or both disease and insect name, or so that's something that we need to talk about.
Yeah. Bob.
So I think we're talking about two different things here. I agree with what Jason and I think what Akif just said. We're talking about naming this complex, this disease, and I think you need to go beyond some sort of shot hole borer for that case. What Tom said about naming it the polyphagous shot hole borer and still keeping the name fornicatus, I don't think ESA would approve that, because the species name fornicatus, if we're still retaining that name, has a common name already. So I don't think they'll accept the polyphagous shot hole borer for fornicatus. So until you get another species name, I think you're stuck with T shot hole borer for the beetle. But I think what Jason said is very appropriate. You need to have a good name for this, whatever you want to call it, disease complex or something that's more than just a shot hole borer. Fusarium, polyphagous fusarium, how about that?
Okay, it's obvious something we need to work on. I do feel that we need to move on, because otherwise we're, one more.
I suggest shot hole tree pox for the disease, but from STP. So from a research standpoint, from a grower standpoint, they're doing a lot of girdling of trees, and they're also injecting phosphorus acid into the trees, and they're doing pruning, and all of these things are wounds. And will these wounds attract more beetles to them? Are we going to predispose some of these trees for an attack?
Well, I think that's definitely a good question. Keep it in mind for a later session, a later part of this session. I think at the moment we can agree, I think, that this thing needs to get a good taxonomic treatment. I would suggest in the meantime we call it Eualacia fornicatus. Maybe we'll call it the California Israeli form. And as a common name, I think we're going to see whether we can get this polyphagous shot hole borer through the official channels. And I hope everybody can sort of agree to that. Otherwise, let's go on to the systematics. What are the main concerns in the fungal systematics? Are there any research priorities there that we feel need to be addressed? Or do we think that at this point we have such good systems, multi-local sequence typing systems in place, that we have a good idea about what we're dealing with and what kind of a variation we have within this particular complex? Wow, okay. I guess the fungal systematics is a lot further advanced. Yes?
As far as the fungal systematics goes, well, I think we have the tools, as Kerry has so well defined them, and he's carrying on with the phylogenetics of the different species. And I think everything is channeled through Kerry, so we should have a very good map of where the species are coming from and where they're fitting into the tree. As far as naming them, we also are on our way. So we've got a very good collaborator in Japan who's doing the morphological characterization. So once that's complete, then we'll come up with a name, at least for the species from Israel and California, which appear to be identical.
Okay, thank you. So is there any action underway to get Fusarium sequenced, or the complete genome, or has it already been done? Do I exhibit my ignorance here? And do we find that whether that would be useful? Yep.
We don't have immediate plans to sequence the genome, but we are moving in that direction. So it's possible that either we would do it in Peoria or have it done through the JGI or BGI or some other place. There are actually a few serial genomes that have been completed for members of the Solani complex. But only one of those is publicly available, and that's through the JGI. That's the P pathogen. But it'll be quite useful for annotation.
Okay. All right. Great. You? Yes?
Before, we have to do a genetic study. Why we didn't do like cross-inoculation? Because of Fusarium, some isolate or some strain is the secondary infection. Mm-hmm. It's not pathogen.
Yeah. Yeah, I think in this case, the status of how eventually the pathogen is interacting with the different host species is still a bit up in the air. Whether, we saw some examples yesterday from Israel where in young plants, the young plants seem to be able to overcome this thing. In older plants, they seem to be causing dieback. And so I think that is an issue that will need to be addressed in a lot of these important host plants. Yes?
Actually, we should study like host strength.
Yes, I think that definitely has an... here, let's say in the urban forest, we don't know what happens to most of these trees that are being infested. And I guess we're having a natural experiment going, and time will tell. But, yeah.
I also think that maybe we should do some population study of the Fusarium in California, compare it in California and Israel, so we can identify if it was where it's coming from. Was it introduced in California from Israel, or vice versa? Is it coming from Thailand? I think this is also relevant.
I agree that at some point is an important question. Since we don't know where, let's say, the natural range of this thing is, I think until we know that, then we can start asking questions about, how did it get from here to there and the other way around. At this point, our beetle work shows that here in California we got 2 haplotypes, and so far in the few beasts that we've done from Israel, there's only 1 haplotype. So if that holds up, then we couldn't have gotten it from Israel, but Israel might have gotten it from us. So anyway, yes, Akif.
Yeah, I just want to make comment on the host susceptibility. So far we know that this complex is like the causing problem on different hosts. So sooner different hosts needs to be screened whether they are susceptible or potentially susceptible.
Okay. is this, I want to go on now to the biology of the beetles. What do we feel that is an important thing to learn there? Of course, we don't know anything at this point about the life history of this thing here, the development rate and all that sort of stuff. Tim, you have ideas.
Well, you just mentioned a number of the really critical ones. The life history of this insect in this environment, number of generations, the temperature tolerances, the reproductive rates, all of those things need to be determined under California conditions. And there's an additional twist on it that has to be part of that consideration, and that's how the host affects the life history, and also how the fungal components, the various fungi that are associated with it affect the life history in those different hosts. So you can't look at each one in isolation. You have to look at the complex and how the components in that complex interact with each other.
Sure, but I mean, that seems to be making the complexity, like you were talking about yesterday. We need sort of a simple things that we should be doing now over the next, what's the priority? I would imagine the priority is to look in some of the hosts that we already know that these things reproduce in, and sort of measure those things maybe in 3 hosts, and like castor bean, California live oak, and avocado. Because then, we have the major hosts at this point, and I think with that, then we can see whether hosts really influence the growth of the beetle or not. Do you agree?
I think you also need to look at some of the potential hosts. I have to make a distinction between trees that are attacked and trees that support development. And so the 3 that you've listed, we know support development. But what's happening in those hosts that are attacked that may turn out to be sinks for the population? And I think we need to spend enough time looking at it to be able to make the distinction between, from a life history perspective, what are sinks, what are sources, and is it a result of a change in the suitability of that plant with relation to the insect, or is it an effect of the fungus as well? So it may be that the host doesn't support the fungus, which means it doesn't support the beetle. And so from a biological perspective, from a life history perspective, it's, I think, important to distinguish between those two. And it may have an effect on host condition as well.
Yeah, we've got about 300 tree species that have been attacked. So
it's a matter of selection.
Yeah, I would say so. Okay.
I was just going to make a comment about that too. I think it's really critical to get some information about the susceptible hosts, how quickly they die, and what sort of the incidence and severity is among the very susceptible hosts. That information, in addition to extending our understanding of the biology of this thing, would also be extremely valuable for trying to educate the public about the impact, potential impact, regulatory agencies and things like that. We had early on some data from some simple plots that Bud Mayfield did on laurel wilt, and he showed the disease progress curve basically for this thing on a very susceptible host, and we still use that data to this day to talk about the impact of it. So just some simple plots, but I haven't seen any real data on the number of trees and how quickly they've died, like box elder and things like that. So that would be, I think, really valuable too.
Yeah, I agree. And let's say we are just starting. So I don't know, 4.5 months. We haven't seen a tree that got infected and died, but we've seen dead trees. So.
There's one other thing that is part of the life history, and that has to do with the issue that was raised just a minute ago, and that's predisposition. Are there predisposing factors that might change the susceptibility of a particular host plant or the suitability of that host plant? And pruning wounds or wounds to the trees might affect the the susceptibility, the predisposition. The watering regimes, we've already heard a number of times over the last couple of days that trees growing under certain conditions, overwatering apparently is one of those, are more susceptible than trees that are not overwatered. So teasing that out is, I think, an important factor that we need to look at. And we haven't heard much about it yet, but fertilization may also be an important consideration in terms of susceptibility or suitability of a particular host plant for the insect.
So as a plant physiologist, I'm happy to hear there's room for us to work on this problem. But in thinking about avocados in particular, because we're affected by salinity in our irrigation water, which would be a stress. We're affected by, we have widespread problems with avocado root rot, although we inject our trees and we do have tolerant rootstocks. That's a stress, and I'd be interested in hearing from one of our plant pathologist friends if there's known to, is it known that a tree that would be affected by Phytophthora would be more susceptible to Fusarium or salinity? Because those would certainly be predisposing factors within the avocado industry, at least, that would make things more difficult.
Yeah, a lot of this is not known. Salinity definitely, we talked about it when you were in Florida, salinity definitely interacts with Phytophthora. I mean, that's been demonstrated with other pathosystems. Fertilization, nitrogen interacts with Fusarium diseases over and over again. Fusarium wilts, the best examples, pitch canker here in California, Fusarium circinatum. Nitrogen there is a real powerful interaction where you have a heavily fertilized tree is going to get hammered much more by that disease than not. But there's just a lot of the stuff we don't know. And to me, it's a no-brainer to think that a tree that's not happy and has got severe root rot or other problems are going to have more problems with a disease situation like this where the vector just loves stressed trees. So to me, this whole thing about what Tim has been saying and Jason, I mean, we need to understand the epidemiology of this thing much better, and all these things interact. I mean, there's not one thing in isolation that we can look at that's going to give us a better picture. We have to look at everything.
Just to add to that, there is some indication that there is an interaction between Phytophthora and Wanlongbing, or the Liberibacter that causes Wanlongbing as well. And there's also some indication that phosphorus plays a large role with Liberibacter as well.
Okay. There. I do want to say something. I mean, we've got to look at all of these things, but what we are trying to do now are setting priorities. So, yes, we need to look at all of these things, but what do we think needs to be done first? That's priorities. I think it was somebody over there who wanted to talk. Oh, yeah, go ahead.
I think for going back to the beetle biology, to try to establish a colony or to have, that will be useful to learn many aspects and for future work.
Okay, well, we already know that, how they rear them in Israel, so we have a little advantage there. Yes, and Mark?
We're really worried about this thing spreading around. Should we spend some time trying to address some very basic dispersal issues from these infested trees? How far can they fly? Is it really recycling on one tree, or is there maybe 15% of the population which are going to abandon the tree and just fly as far as they can until they find another host? Some of that information would be really useful, especially if we start picking up satellite populations.
Well, I think what Mark said is really important, but it also gets to this issue of can we develop a suitable attractant so that we can trap it effectively. So trap technology, including developing an effective attractant that would make trapping effective, is an important consideration that we need to look at. Right now, there's nothing that we know of that seems to work very well. So that's an area of study that I think is going to be critical.
Yeah, I think that's an excellent point. And we'll talk a little bit later more about that. Now, are there, at this point, I think we've...
Can I say something?
Yes?
You know, we keep forgetting that where the source of money is going to come for all these things. And avocado A is one funding, and the other one is the ARS. And she made it very clear, Deb, that a commodity is going to more likely get funded by them than something that will have to go to the Forest Service to get money from. So therefore, in whatever thing that we're planning on doing, avocado got to be part of that system if we want to get funds to be able to deal with it.
Okay. Yes?
Which I'm going to reiterate what I said yesterday. I think if we can have a dollar figure or some kind of figure, I think that speaks a lot to grower, to the eyes of the grower and to the ears of politicians than any scientific data that you can show. So if you can come up with some kind of figure that shows like how important the disease is, then that would be helpful for the future.
Well, I guess, you know, at some point we can do is we can make estimates of, you know, street trees dying, and removal of a street tree is very expensive. And then eventually, if they get into, let's say, avocado groves, they're coming up with a monetary value of the loss will be a bit simpler than, you know, determining how much it costs if it gets into the forest. But yeah, we can come up with those values, I'm sure, because we know we've got contacts with people that know in Orange County, I believe, every street tree. They can, you know, give them the species name, and so we can figure out whether these things are susceptible and eventually will die, like California live oak. And you know, just the removal cost for that will be terribly high. Okay.
You may also be able to extrapolate some data from Israel and their experience. If they're, you know, 10% yield loss or 30% loss and this and that.
Yeah. Okay. I think it's time to... Okay, are there any other issues that we need to know about the life history of the beetles? Or have we pinned down all the priorities by now? I think one of the things that is also probably important to know is whether those additional fusariums that Aki found and the gnathium fungus, whether they at all play a role in the beetles' survival, because that, in a sense, does differ from Israel. In Israel, they don't have gnathium and those additional fusaria that you found. So that's something that we could look into to see whether these things actually are playing a nutritional role or whether they're just contaminants. Okay. Everybody keeps an eye on what Joe is typing up there. Joe, type some insults to everybody. See if they pay attention. Okay, so if there are no more high priorities that people see in the beetle part of the beetle biology, then I'd like to go on to the biology of the fungal symbiont. What are our priorities there? I would imagine that trying to find out, indeed, like was suggested earlier, whether the failure of the beetle-fungus complex to start on a particular tree is caused by the inability of that tree to harbor sufficient growth of the fungus. I think that'd be an important part. Any other aspects? Yes?
The basic requirements of the fungus, because if it relies on the fungus for food, where the fungus can grow is going to play a big role in where the beetle can go. So moisture requirements, temperature optima, stuff like that's a good basic place to start.
Okay. Yeah, and we've already seen that the beetles seem to prefer the north side of the tree, and where we think it probably has to do with temperature and humidity, that they like that better. Yes, Randy.
Oh, so I would suggest also to check to see if there's any other alternative modes of transmission, perhaps pruning as a possibility. We've looked at that with laurel wilt a little bit, and because in avocado groves, I don't know how much pruning is done, just standard pruning here, but we have a lot of that in Florida. But just all the urban trees that are going to be trimmed, people are going to be out there trimming branches, and so the potential for movement on pruning tools is, I think, rather high, because if you look at fusarium wilt on palms, that's a huge issue, and it's how the disease has been spread to new areas and things like that. So that's a pretty simple thing, but it could be done rather quickly to look at potential transmission on pruning tools. But
yeah,
it's I guess that's partly management too, but it's part of the biology, I suppose, as well.
Yeah.
I just wanted to focus on diagnosis and management. These are two really important things, and this diagnosis thing is going to be a little bit muddy until we understand which Fusaria are actually doing damage. So that, right out of the gate, what do we find in the beetle? What do we find in affected trees? And which of those Fusaria are actually causing damage when we put them back in the tree? That's really important. There are a lot of other Fusaria out there that are probably going to muddy up the picture until we understand that. Once we understand that, Keith has got a diagnostic for his one Fusarium. That's a good start. But again, the caveat that we heard from Dave Geiser and Carrie about, don't put all your eggs in one basket and assume that diagnostic is going to go ahead and let you know when you have a problem always. Just keep your mind open on that.
So, so do you think that a multiplex kind of thing, where you go for several different genes of that particular fungus in your real-time PCR would be a better approach? Because if you will only call it that fungus if let's say all three genes that you've done amplify with the particular melting curve or so that you expect.
Yeah, I think the first thing to do is make sure that you've got only one Fusarium involved. There might be additional ones. From what we have so far in Florida, it looks like there are two that when we put them back in avocado, they perhaps cause symptoms. So, we need to understand that first, and then when we understand that, how you go about identifying those Fusaria, whether you use TEF, which is what he's using, or you use microsatellites with Jason's using for a RAF. I mean, that's another discussion later on about how to best identify those. But first of all, understand what those pathogens are.
Okay, but so in Israel, you just have one?
One clone.
One clone, okay.
Yeah, but then the management thing, and that's going to be the tough one. I mean, based on our experience in laurel wilt, this is not going to be easy. So, understanding all this stuff, and Jason brought up a really good point about, moving by pruning shears, cultural, chemical, and resistance-wise. I'm not going to hold my breath on resistance for laurel wilt, and it sounds from Zvi yesterday that there's probably not real good resistance in avocado either, but if we're talking about avocado only, let's understand that. Mm-hmm. And then chemicals, I think what we're thinking now with laurel wilt is identify quickly those trees that are affected, get them out of the circulation, and then treat surrounding trees to avoid root graft transmission. Is that going to happen with this problem? Doesn't sound like it's going to happen, but let's understand, whether that's a possibility. Is this thing going to move within a tree? Is it going to move via root grafts? I don't think that's understood yet.
Yeah, and I think here it's also, and I'm not quite sure what exactly is the case, but the fungus is not quite as virulent as the one you guys are dealing with, and so simply being infected. If you just infect an older tree, you get dieback. That's what you guys have found in Israel?
Yes.
And the dieback happens, how soon does that happen?
Probably about a year.
Okay, so no fungus welcome there.
Yeah. Can I make a comment on the Fusarium? So in our case, the Fusarium new species is the most common one. So we found all new Fusarium species on all of the hosts. Those Fusarium solani that we found were the additional in the same host, and then we found the different solani species. But the Graphium is the anamorph of the Ophiostoma group of the fungi, so they are mostly associated with the Ambrosia beetle. So that's a very good point that we need to find out So what is the role of that graphium in terms of the food for the beetle? And we still don't know that graphium is the pathogen or not. So that's the first thing that we are going to do. We are going to do the pathogenicity test with those fungi to find out if they are whether pathogen or not.
Okay, question over there. Yes?
Yes, I wanted to go back to the comment that was made just a minute ago by Randy about chemicals, and one he made earlier referring to the National Plant Disease Recovery Plans. The NPDRS has run out of ARS, and one of the things that if a chemical that is not labeled on the crop is identified as being useful in managing the disease, ARS will work with EPA to get a Section 18 exemption for that chemical, so that you have those in place before you have the problem. And so it might be to your advantage to try to get this into the pipeline for the NPDRS plans.
I think that's an excellent idea, but my impression from what we heard yesterday was that there were not really too many chemicals that were going to be working. But I think it's something we definitely need to emphasize and hope for the best that we can find some chemicals that actually get into the tree and protect it. Yes?
Yeah, you're going to want to, if you're going to try to get a Section 18, you're going to first need to survey different chemicals, get some data on efficacy, and it would also be a good idea to get some preliminary residue information. So it takes a little bit of time, and then you also have to find, there has to be three things on board. The owner of the chemical has to agree to the Section 18, and you have to have the background data, as I just mentioned, and then you have to have EPA's acquiescence as well.
Okay, but I mean, it would be a high priority to have this. What's the estimate of getting this? I mean, of getting a chemical that will work?
Yeah, well, I can tell you, from the time you submit your application, supposedly, if you submit your application, EPA has 50 days to respond.
Okay.
But it's usually not 50 days.
I mean, they say no immediately, or?
Well, it's called a Section 18. It's an emergency exemption. They do have a crisis exemption that involves, both of these involve also your state regulatory agencies getting involved as well. The crisis is a little bit faster, but usually states don't like to use crisis exemptions unless they're really comfortable with it. So, when you say emergency, it's a relative term.
Okay. Any more? Somebody? Yes.
Regarding chemicals, Ruben passed to me this. Jim Atascavage wrote an email to Stanley this morning. Jim could not be here today, but on the Riverside campus, Jim Atascavage is probably our expert on fungicides. And so Ruben's asked me to read this to you. Basically, it's about SDHI fungicides. So he says, I suggest evaluating the SDHI fungicides as a FRAC group 7 because of their reported systemic action and movement in the xylem. These include, now I can't pronounce these names very well, but fluoropyrum, which is Luna privileged by Bayer, fluxaperoxide, which is a BASF material, and possibly penthiopyrade, which is a DuPont product. Many of these fungicides are pre-mixtures. That's based on your studies. Fluoropyrum and tebuconazole would be likely one to test along with possibly a tank mixture of fluoropyrum with metaconazole. Others include a pre-mixture of fluoroperoxide or fluoropyrum trifluorostrobin because they have an SDHI and a QOI component. I am suggesting mixtures to minimize selection for resistance. One idea would be to find the most systemic DMI and tank mix it with one of the systemic SDHIs. Another idea is to choose a similar DMI imidazole fungicide chlorchloraz, which is non-registrable in the U.S. due to toxicity, and evaluate this alone or in combination. One such DMI imidazole would be trifluomazole, and again, could be tested alone or in a mixture. And all these materials are registered on tree crops already in the U.S. with MRLs in many countries of the world. And then he says, I disagree with Ruben. I think all the fungicides that I have mentioned have the potential to be registered on avocados in the U.S. and in many countries of the world.
Okay.
Okay, none of these fungicides are available in Israel to date. So we'd have to test them first in vitro to see whether they have any inhibition on plates. Thereafter, we'd have to devise some kind of method to determine their efficacy in infected trees. So obviously we have a lot to do before we get to this stage. And again, none of these fungicides are currently available in Israel, so I don't know if in the long run they would be effective if we'd be able to register them.
Okay.
A little bit of warning on the QOIs. We tested those in vitro against our pathogen, RAF. They're highly effective in vitro. They don't do squat in the plant. I don't know if we can't get them in trees or what, but I've talked to other people who have worked with other fruit trees, and you just can't get the strobies in trees for some reason. So I don't know what the deal is with that. Strobies would be great. They have a totally different spectrum of activity, but you can't get them in trees. So in vitro activity and in vivo activity are often two totally separate things.
Okay. I think okay.
Jim suggested QOIs.
So one thing I want to ask is if, let's say, if a tree's xylem system has been compromised by these beetles getting in there, is that, what causes these trees to be unable to move all this stuff up into the tree?
Yeah. One of the things we noticed early on, first of all, for our system, and it may not be, what you've got here, but when we see external symptoms of our disease, the inside of the tree is hammered. Mm-hmm. We have a 1 to 10 subjective scale, and when we have a 2, one notch above healthy, inside of the plant is a 5 or a 6. Mm-hmm. So that's unfortunate. And when the take-home lesson on that is when you have a lot of internal symptom development, those pipes, those vascular elements are full of tyloses and are non-conductive. So you can add the most effective fungicide or insecticide to that tree, and it's not going to travel because the pipes are plugged.
Yeah. Okay.
Just a quick comment on the chemical.
Because I've been told to hurry up.
Okay, chemical control of it. So this meeting is reaching its goal actually. So we got the funding from California Avocado Commission to work on the control aspect for the disease part of this complex. So all these chemicals that was mentioned that we are going to try and in vitro first, and then we are going to take them into the greenhouse in the second. So thank you very much for all your comments and suggestions.
Okay, so let's now go on to the monitoring and control strategies. And so we obviously don't have a really good way of capturing these beetles yet. We have been inspired by the work they've done in Florida, in Georgia, where they used these bolts hanging up in the tree in order to see whether particular tree types would attract the beetles. And we've talked with Tom Coleman from the Forest Service, who's going to get bolts, and with Huntington Garden, where we have a place with lots of beetles, and we're going to try out to see whether these beetles are actually attracted to a particular tree. And then hopefully if they are, then we may be able to, with the help of maybe Jocelyn, figure out what is in these trees that makes them attractive. And so, but any other ideas about coming up with an effective way of monitoring these things? What Akif and I have been doing in our surveys is we just drive down streets and we look at the trees. And the symptoms are generally so obvious that you see big wet patches on the trees, and you can say, okay, this area is infested, and you drive a bit further down, and you find fewer infestations, and in the end you don't find anything. And then we say, this is the edge of the distribution. But it would be nice to have a much more efficient way of, let's say, capturing beetles. Yes?
One thing you may also want to employ is if your Department of Agriculture has a monitoring division for fruit flies and other pests, exotic pests, is what they've done in Florida is the people in the field, the inspectors in the field that have the traps, set the traps, they add on to it that they select sentinel trees. So let's say the box elder. And so on their route, they have selected trees that they monitor on their normal rounds. And so that increases the number of eyes out there looking and reporting.
Yeah, I think that's an excellent idea. The intention is too that around avocado growing areas, the people that do the pest control there are going to keep their eyes open, and Akif will train them. Yes?
So there's another point. This is actually Ruben's first idea, fungal volatiles. So we also need to look for what is the importance of the fungal volatiles, if they are attracting the beetles or not.
Okay. Okay. Any other ideas about monitoring and then control strategies? What are our high priorities there? Nobody has any idea. Yes, an idea.
Well, I don't know if it fits the category, but on the control aspect of things, I think we should understand the mechanism of resistance in the tree better. Trees are known to compartmentalize the invasion of fungi or the insects. So the tree is responding to the invasion. And I think it would be important for us to understand this a little better. There's a poster here, actually, that's showing some of that with laurel wilt disease. So try to understand why trees are resistant or susceptible to a particular disease by looking at their defense mechanism, like tylosis or cell wall compartmentalization, lignin chemistries, and so on and so forth. I think understanding mechanism of resistance will help, I think.
Okay.
I just think as far as the control, we've seen a lot of evidence with the chipping working in, Sri Lanka, and also with Jason's work with red bay ambrosia beetle. I think that's probably going to be the main resource for a lot of these arborists to sanitize trees. And I think if somebody's going to start going down the avenue of in dealing with infested material, chipping is probably the way to go. We just really need to see how effective that is.
Yeah, that's one of the first things Tim is going to do when next week.
As I said yesterday, there's a number of things that we're going to be looking at, and chipping or grinding, reducing as part of a sanitation program, solarization as part of a sanitation program, what to do with the wood that's infested, and we'll also be testing insecticides, much like what has already been discussed with the fungicides. So we need to look at a number of other materials that we have some good data from our colleagues in Israel, but we need to do the work in California as well. So we'll be looking at a variety of different materials on a number of different hosts, and look for ways of potentially managing them on a short run, looking at controlling populations in particular environments. So hopefully we'll have a number of things in place fairly soon for short-term control. A lot of the other issues that we've talked about are more long-term approaches.
Okay. Mary Lou. Oh, Jocelyn.
Just a question, since grinding has come up. Does anybody really know whether these beetles get down into the root collar and the crown?
We
they do?
Yeah.
Okay, then grinding would be probably necessary.
Mary Lou? Oh, question back there. We'll take that one first, and then we'll go to Mary Lou. Yes?
Regarding monitoring, I'd like to go back to monitoring just so Joe can write a negative comment about me, but double-sided sticky tape on some of these heavily infested branches that we saw at the Huntington Library where these beetles are crawling or flying and hitting things, and it would be a static way of maybe keeping an eye on when the population is going to take off or explode. We could count the numbers.
Yeah, we've been doing a little bit of that where we've looked at those trees. We've hung up traps, and we saw when we went to the Huntington this last Sunday, we saw the one tree where we left the big old purple sticky trap for, what was it, 1.5 weeks or so? And this thing was just completely covered. So if you hang it up on infested trees, you can catch a lot of beetles. And but to know when they are ready to fly, we don't know the seasonality of these things and whether they have seasonality. What I just found out, and I was really happy to hear, is that the CDFA has been running traps in the Huntington for years, and they collect them every 2 weeks. So we're now together with the CDFA going to look at, okay, when did this thing arrive there? What kind of a seasonality is there? So maybe we'll even be able to get an idea of how many generations there are per year. So, but on individual trees to tell when they're ready to go, I don't know. Yes. Any other monitoring? Yes.
Well, this is sort of goes on both, and I wanted to go back to the comment made on tree bolts. I think that's a really good avenue to go, especially on avocados, because we have a very nice germplasm collection. We have a mapping population that represents a lot of the genetic diversity that we have in our main cultivars grown in California, and I think if we could take advantage of the mapping population and the germplasm collection in terms of looking at susceptibility between different varieties, I think we might be able to really learn something and come up with some ideas that could feed into volatile emission of the tree or the response to the beetle and the fusarium boring into the tree.
Do you have old trees to turn in for that, or only young trees? Yeah, but you don't want to use those for infecting them with a fusarium, I would assume. Okay, all right, okay, great, yeah.
Just going back again on the monitoring, if you start to put out traps in an urban area, it would be a very good idea to get immediately the media involved. With our experience with ML ash borer, when we start putting these blue sticky traps, these purple sticky traps in the urban environment, in streets and things, it's when really the people, the city took notice, and people really started to start to report and look for beetles and things like that. So I would say the moment you start putting out even experimental traps, get the media involved. It's a good way of getting the message across.
Okay, yeah, thank you.
Yeah, this is nothing to do with monitoring, but mainly looking at the plant health. We knew that when the plant is under stress, the crop is susceptible to the pest attraction or damage. Why don't you look at the cultural practices, especially with respect to fertilization or using ample ratios of nitrogen and other nutrients, perhaps coupled with monitoring water irrigation levels, right? Looking at precise agriculture agriculture practices.
Okay, thank you. Okay, Ruben.
I like to suggest that we can just look at fertilization, at how it affects necessarily the beetle, that the effect on the quality of the fungus could be even greater because the beetle is feeding on the fungus. So you can't look at fertilization separate from the fungus. So if we say, oh, well, we got better fitness, well, what about the fungus then?
Yeah. Yes, over there.
I suggest also to follow up on the idea of Beauveria bassiana as a control measure, especially link it with the shipping experiments that you're going to do to attract. Sometimes when you ship the trees, the infested trees, that attracts more trees, or if you find an attractant, to combine that with Beauveria control.
Yeah, we will definitely look, I think, into Bavaria, but for some reason we don't stand up here and be really enthusiastic about Bavaria because it's generally really hard to get going in systems, and it might work really well in really moist environments, but in dry weather these spores have a really hard time of germinating. So we'll look into it and we'll hope for the best. Yes, Diane.
There is a product out there called Botanigard, which is a commercial formulation of Beauveria. And one of the formulations is in oil, and it has sunblock and all this stuff. And it just hammers scolatine beetles. Okay. The nice thing about it is it's compatible with just your standard application equipment. You know, you don't need anything special, no agitators. So you might think about playing with that a little bit. I mean, it has its drawbacks too, but it's considered safe, so you can put it in places like Huntington Gardens. I mean, it's registered for organic cropping systems.
Okay, that sounds great.
So, I mean, it's a pretty good product, and we've played with it with bark beetles, and it's incredibly strong. And what we found also, which was kind of shocking, is the beetles that are predators on the bark beetles are not susceptible.
No? That's marvelous.
You don't get that lucky every day.
Yeah, no. Yes?
Just I want to mention that indeed our ambrosia beetle is extremely sensitive, susceptible to Beauveria. And in petri dishes, they die very fast. However, application of Beauveria in some preliminary trial was a complete failure.
Did you use this particular, oil mixture where they really protected the spores from sunlight and drying out?
Well, we did it in what we thought is the best environment we could find for Beauveria.
Okay.
And it was a complete failure.
Okay.
But these are preliminary tests, so
we'll try it. Yes.
Just another thing that we're looking at in citrus, going back to the volatile organic compounds, where there is very good reports that you can detect whether a tree is infected with Spiroplasma citri, citrus tristeza virus. They're working on citrus canker. They're also working on citrus variegated chlorosis, and of course, one lung being. The equipment is the actual sensing equipment is it's already gone to a manufacturing company that they're looking at making a nice like handheld equipment that can be easily deployed in the field. So, that may be another avenue to explore with this pathosystem.
The electronic nose, huh? That's
the electronic nose,
yes,
correct.
Yes, no, that works marvelously.
This might be a little bit of a long shot, but since we're just kind of brainstorming here, with another fusarium disease, pitch canker, which is on conifers, there's been quite a bit of work demonstrating systemic induced resistance in hosts to that pathogen, and they've demonstrated basically that one infection basically can lead to further resistance down the road. And it might be worth playing around with this system, because it seems like you need a lot of infections, obviously, to result in death to the trees. It'd be interesting to know if you went out and pre-inoculated trees and then kind of observe the natural infections that take place if there's any impact. I mean, it's a long shot, but it has been demonstrated in another fusarium-caused disease.
All right, thank you. I think unless people have a real high priority issue in this case, then we should go on to the epidemiology. What are our priorities there in addition to determining all these life history characteristics? Anybody, any thoughts? Matt, so thoughts. We need thoughts from you.
I'll give you my wish list at the end of my talk.
Okay.
And so the obvious ones, and I think it was touched on a little bit, are understanding what fraction of the beetles really are carrying Fusarium at any given time. If it's super high, then it's sort of an interesting situation because it means you can't really separate the disease from the insect, and that certainly impacts things. So getting that is a really fundamental piece of any understanding of epidemiology. And then looking at some of the other things I talked about, and have been touched on by several people, relative differences in sort of susceptibility, resistance, tolerance, whatever, to the pathogen is certainly something that needs to be looked at as well.
Okay. Yeah. So I think you, Randy, you mentioned that not everybody was infested with these symbiotic fungi. So what happens when those poor deprived beetles without their symbiont hit a tree?
Well, get away from the idea that this is the only thing they eat. I mean, if they don't have, the Fusarium you're interested in, or if they don't have Raffle-Auricula, there's other stuff in there that they have in their mycangium that they will consume.
Okay.
They're not wired into only eating that. So just because you don't see Raffle-Auricula in one of these individuals or your Fusarium, they have other stuff that they can fall back on. Now, that said, in our system, Xyloborus glabratus, I mean, Raffle-Auricula prevails. It's usually there, and when it's usually there in high numbers.
Yeah.
One thing I'll add to the mix here on this, though, is that we've identified what we think is a threshold. In red bay and avocado both, very sensitive pathosystems, we can kill trees with 100 conidia, but below that, I think we're getting away from that point. So if you want to look at glabratus or some of these other scalotines that we're looking at, you get below that 100 for most of those individuals, for most of those species. And in some of the no-choice stuff that Daniel talked about the other day, we've seen, it doesn't happen often, but we've seen examples where infection occurs, but systemic development of disease does not occur. So two things to keep in mind with your system.
Yeah.
You might have a situation where you get infection but no disease.
Yeah, well, it seems to me that maybe our system is a bit different since, let's say, the Israeli beetles couldn't survive on the Fusarium from Sri Lanka and the other way around. So there does seem to be at least some specificity.
But understanding what your beetle's doing definitely is, boy, if you can figure that out, it'd be very good information.
Yeah.
Yeah. From what I read, it seems like the differences between hitchhikers and phoretic and gut fungi play a big role in this thing. And somehow or another, we have to learn how to separate where this, because if it's the case that we require so many, such a count, and if the count is not arrived at, we cannot just look at the mycangial complex and say, well, it's right there, and therefore there's so many spores, and therefore that's not going to be a big deal. However, maybe other parts of the body can bring on additional, and then it's going to. So how you study this, where the source is coming from, going to be very important.
Yeah, but I think that at least initially we can do these experiments like they did in Israel, where you just grow a particular fungus on a plate and you see whether the beetle can survive, and then in the end you can do a whole series of them, like the ones that Aki found, those other Fusaria, can they grow on that? Are they real symbionts? And you can make mixtures and whatever. Yes?
Diana should be talking about this because there's better examples with bark beetles, but perhaps with ambrosia beetles as well. You don't have to have a nutritional symbiont be cause for concern. Sometimes you can have a phoretic associate. The Dutch elm disease is the best example, where that pathogen it's not a mycangial thing, it's on the exterior of the beetle. The beetle doesn't like the fungus, but the fungus still kills trees. So,
Right. Yeah.
yeah, so Ruben's point about understanding what the ecosystem on the beetle is a good point.
Okay. All right. How about predicting the spread of this beetle, where it will go, climate matching? What do we feel that's a high priority? Or Ruben thinks it's a high priority?
It's a highly important priority because I think we need to learn about flight patterns and also wind patterns, because you're going to get movement by wind. You know, we have this. We cannot take all the time the Israeli experience for granted for us, because they have their coastal Mediterranean high humidity during the summertime, which is definitely not what we have. So therefore, we can get into a lot of troubles if we think that we can duplicate the Israeli experience.
Yeah, but I think Tom is already going to do some climate prediction with the zones that are infected in California at this point. I think Tom disappeared. There he is. So what are you going to do with predicting where this thing is going to go?
You'll be the first to know, you and I keep.
Yeah, yeah.
So, I mean, I think, sort of the practical aspect is set up, and I'm actually drafting a little sort of proposal among ourselves, is to set up rules and the repository, so as soon as we get any kind of survey data from any means, that's what is going to be the basis for making any predictions later, so that we can keep this and available to whoever the owners of data sets are willing to let see it. This is not hugely expensive to do. And of course, the more samples you get, the better the accuracy of the predictions becomes.
Yeah, but of course, at this point,
of course,
with the
like
spreading,
anything else with funding, it goes a lot faster.
Oh, the spread goes faster, huh?
There's other things being proposed. This is a relatively inexpensive one, so it's a real bargain.
Yeah. Yes. All right. Okay. Well, any other priorities? Forest Service, the natural areas. You guys are on the ball. You're checking. You got it covered. Yes. Okay. Great. Well, are there any other things that haven't come up in all these different sections that you think we've forgotten about? How could we bring it up now? Yes.
Well, maybe it's a little bit tangential to this, but one thing it seems to me, I'm not a plant pathologist or entomologist, but I've been observing the interaction of people here, and I mentioned it to Deb Fravel a little bit earlier this morning, is that within the USDA structure, there's a possibility of starting a working group or whatever that maybe down the road could allow people working on invasive beetles to write a joint grant or an avenue to continue communication like we've started at this meeting. So maybe Deb can make a comment.
Isn't there already a working group being assembled? Leslie? Working groups? Yeah?
Well, the new pest advisory group is looking at it within PPQ, and so we'll share that information with the emergency and domestic programs and other people within PPQ. CDFA is also involved. We have a state plant health director in California that's involved, and so kind of a it's not an official working group, but it is with its, PPQ is looking at it to see if there's anything that that we could do.
So that's all that's the group where we have these conference calls.
That's the group where you have. I think CDFA has been initiating the conference calls, and then California PPQ from the State Plant Health Director's Office, and then CFIST, which is our group, and some people from the Western region have been involved. And so what the step right now that we're in is just putting together a report, and that's why I was given permission to come here to get the best, most current information on what's going on, to see exactly what the situation is and whether there's something that can be done from a regulatory perspective.
All right.
I would just like to point out that one of the most successful working groups that I have ever seen anywhere is the HLB working group, and I'm sure Mary Lou Pollack can tell you a lot more about this than I can, but it truly is an interaction with industry and the state and the federal partners and all the parts of the federal government. We have it organized under APHIS, but certainly there's huge participation from ARS and from other parts of the government also. And what they have done is divided up into subcommittees, and they set their own agenda, kind of like what we did here with the outline today. They do monthly calls with those, and then the leaders of those report together, and then they meet once a year to assess where they are and find out where they should go from there. But I haven't seen any duplication of work. I've seen people working together extremely well, and they were able to leverage some very modest initial amounts of money into some very serious funding today by working together and getting results.
Okay.
That is, even though there's not at the moment an official working group, the information that I've gotten, I'll pass on to the national science program leaders within CFIST, and then they can go on and also make a determination. And so like Deb is a program leader for ARS, our PPQ has national science program leaders, and they'll be made aware of the situation, and they weren't able to come, and that's why I was sent.
So would it be helpful if, let's say, commodity boards here would start contacting the USDA?
No, I don't know. I'm not sure what the proper course of action is. Normally, I think the state plant health director would be the first contact with people within their state, whether it's commodity people or individuals and that sort of thing. So I would say you may contact the state plant health director, and our national science program leaders are aware of the situation. They've been involved in some of the conference calls. They'll be made aware. I'll write a report for the new pest advisory group, but also a more detailed report on what the visits, the pictures, the things that we've done here and what we've talked about here and what the plans are, and then present that. I'll give that to them, and we'll see what happens. And so I'll give all your contact information as well.
Okay.
Okay.
All right, thank you very much. Anybody else who wants to bring something up that we've forgotten? Ruben.
I went out, so I'm not sure. Was anybody mentioned the Pacific Rim possibility of getting funding with people being here from Sri Lanka and India and
the UC has a small granting program for cooperation with scientists in the Pacific Rim. The conditions for that are often very specific, and I think it's more in the social sciences, as far as I've browsed from that from those announcements, than in let's say agricultural sciences. Is that true? Yes. Yes? Okay. All right. Okay, now, Joe has made a long list, and I guess what we could do is just go and have lunch, or we could go and sort of look through this list and see if we find that we want to maybe prioritize our priorities. I think that's probably going to be a rather complex thing, but let's see, how much time do we have? We have 12 minutes. What do you think, Mary Lou? You look troubled.
It's a long list of.
Yeah. Well, we can also, yeah.
The other thing is for us as a group here at Riverside, work on it.
Yeah. Yeah, I think that may be a better idea, because otherwise it's going to be a rather unmanageable affair.
We can send out a vote by email.
A vote by email, yes. Only one-time voting, okay? All right, I think, oh yes, Akif.
If you are done, can I make an announcement?
Okay.
There is a postdoc position available in my lab. If you know somebody who will work on this issue, please let me know. Thank you.
Okay. Thank you very much. All right, I think then with this we're going to go and eat our lunch, and we'll see you all this afternoon.